diseases. And some of the stuff I'm going to skip through. Again, we got we're running a bit behind. But again, let's just try to keep it really interactive. Let me just remind myself what the schedule should look like here. Okay, good. So there are a lot of diseases that affect strawberries, but this is what I tell most people: plant diseases are an exception. And you say, well, that's not true. I know every plant I grow gets a disease. But what I mean is, diseases only occur on certain types of plants and only under certain conditions and only at certain times. So if you get, sometimes people say, oh man, you know what, I had fusarium wilt in my tomatoes, that means I can't grow watermelon because it gets fusarium wilt. That's not true. The fusarium wilt that hits tomato only hits tomato. It's very host specific. So it's an exception when a pathogen affects many different crops, and also with strawberry diseases, we might get some of them year after year. But when I speak, sometimes, especially newer growers think, oh, I don't want to get into strawberries because they get so many diseases. But most years, most growers do an excellent job managing it. So I want to preface it with that. Then I highlight here in yellow just some of the most common ones that we run into here in the southeast, and I'm gonna talk about several of them. We already talked about black root rot and the fumigation we do for that, or farming systems management. The other key thing I think when we're talking about diseases is just making sure we get the diagnosis right. And most states have really good diagnostic labs or extension services. And when I approach a problem, I'm always thinking, as I approach that problem, I think, okay, is this a living factor or a non-living factor? Right? That's going on. And if it's a living factor, then I try to figure out, okay, is it a pathogen, an insect, or a weed, or maybe a deer, whatever. And then, of course, if it's a pathogen, then, it's a fungus, a bacteria, or a virus, and we heard a lot about viruses and nematodes already. Or when we get a sample in the clinic, right away, we take the soil, we look for salts and pH. And we just rule that out as a possibility. And so it's really important, and I would say 50% of the time we get a sample, it's not a disease. It's an abiotic problem, so drought injury or lightning injury or something like that, right? So when a grower calls me and says, Frank, you got to come to my field, something happened overnight and it's really bad, then usually I'm thinking, okay, it's not a disease, because disease tends to have a certain pattern. It happens over a certain time period. And so a lot of times when I'm doing diagnosis, just by asking the right questions, or when you submit a sample by filling in the form really well, that really helps a diagnostician clue in what it might be. So diagnosis is key. These are some of the ones that we're not going to talk about. Leather rot is caused by a Phytophthora hissaberi. I rarely see it in the southeastern part here, but if you get it, it is terrible. Even a little bit of disease makes the fruit taste terribly bitter. So it turns off your customers, but I rarely see it, okay? And then the leaf spots, I won't be talking about the leaf spots, powder mildew and common leaf spot. I won't be talking about that. There's the powder mildew. But, we can go back and forth if there are specific problems. The most important disease we see year in and year out is called gray mold, caused by Botrytis. And especially when it's cool and wet. So this spring has been, very favorable for gray mold. We've had a lot of cool weather, wet weather, and there's a few things to know about its biology. Napoleon said, know your enemy. And the more we can understand the nature of our pathogens, the better we can control them. So Botrytis, the most important thing to know is in this life cycle here in this picture. Here you see some strawberry tissue. This is a petiole, and this petiole is dying. So what happens is the botrytis often comes in on the plants, or it comes from the surrounding fields, and it grows on the green leaf, and it just sits there. It's called a quiescent infection. Then when that tissue dies, then botrytis grows all over it, and just look at all that gray fuzz. Well, if you hit that, if you touch it, you would see a cloud of spores come off of there. Okay, so those spores then, they go into the flower in the stamen, and then they grow down to the stem end of the fruit. And then the botrytis just sits there until the weather is right. And then as the fruit ripens and the weather is right, then it expresses itself. So here you can see the brown rot occurring from the stem end. 90% of the botrytis you get in your strawberries comes through the flower. So that's really important to know. That means you win and lose the battle at flowering. You don't see the rotted fruit until about 24 to 26 days later. But you lost the battle. You've lost the battle 3, 4 weeks ago. So that's really critical. And I'll come back to that. About 10% of the time you see this fruit here, it's rotting from the bottom end up. But that's because it's sitting on dead leaves full of botrytis. So botrytis has a foothold, and then it can grow in the tissue. And it's called the necrotroph. So what it does, it just produces all kinds of stuff like enzymes, and it just breaks down the tissue, and then it feeds on the tissue, the strawberry tissue that's breaking down.
And does bee give off spores as well?
Bees can transmit, I suppose, but it's not a big problem, not for strawberries. Unlike if you grow apples and you have fire blight, then bees are very good at moving the bacteria from one flower to another. And every flower bud might get fire blight. So that's where we see it. Now, if you have bees that are visiting flowers, what some people do is you put in front of the hive a beneficial fungus called trichoderma. And it's formulated and encapsulated. So now you have a bee delivery system that brings your biopesticide right to the flower. Okay, so there's some, once you understand the biology, and you lose the battle in flowering, so you got to protect that flower. So there's some biopesticides in bee work that's been done. Yeah. So yeah, great question. So that's the most important thing to keep in mind with botrytis. So that's why when we give recommendations, start sprays at 10% bloom. That's why. And I'll walk through some of the data we have that shows how knowing that biology can really save on control. So here's another example where flower parts fell on the strawberry, and then the botrytis gets a foothold. But that doesn't happen a lot. Most of the botrytis comes in from the stem end because it got started in the flower. And we've done a lot of work just looking at strawberry growth. So if we plant in October, we harvest in April and May, then we can find a lot of botrytis coming in on the strawberry leaves. And this fungus has got it figured out. About 90% of the leaves that come in the transplant died between November 15th and December 15th. And then these new leaves were growing, and they picked up botrytis. These leaves overwinter. They die right when the flowers come out. So the botrytis gets into the flower, and it gets into the fruit. So we could control here, but I would never ask a nursery person to try and control botrytis, because it can't be done. You can't do it on your farm. Botrytis is everywhere, is how you need to think about botrytis. So the key weak link is right here at flower formation and protecting the flowers. Sometimes we see it here in the southeast. We get this botrytis crown rot. There's two reasons for that. One, especially like a cultivar like Sweet Charlie, it'll put out early flowers like in January. And then we get a cold spell, the flowers get killed, the botrytis grows down the flower, down the peduncle, and into the crown, and then we get this rot. Or we have a strong canopy, a very dense canopy, and then we see the botrytis growing in the crown rot. We had a lot of crown rot this year, more than I've seen in most of my 18 years here. But again, we would have specific recommendations for that, and I'll come back to that too. We call it crown rot, but it doesn't really get into the crown very much. It just kind of colonizes all on top of that crown. Okay, so if we talk about botrytis management, I have this as a handout. Just make sure we don't over fertilize the strawberries. Some growers really push the nitrogen to them. The bigger plant you have, the more dense it is, the more botrytis you get. So the way I think about it is grow your crop for optimum yield. Because I know that if you grew 1 plant per acre, you would have a low botrytis incidence, right? But that's not going to be profitable. So you got to grow for yield, but just don't over-fertilize, because they love succulent plants and they love soft berries. And when you're pushing too much nitrogen, you're going to have soft plants. If you're organic, removing dead and dying leaves just before first bloom, that costs about $300 to $400 per acre. But a lot of growers will do that, and they'll go through and they remove the dead and dying leaves, because what else are they doing? They're pulling the weeds that are coming out the holes. A bunch of the plants will be growing under the plastic, because over the winter they put out their shoots and the new crowns, and they might be under the plastic, and so they pull those plants above the plastic. So they're doing 3 or 4 different things when they're going through the field like that. But like I said, that's about a $300 or $400 per acre job. And you can substantially reduce the amount of botrytis that you get. But if you're a conventional grower and you're going to use fungicides, it probably will not pay for itself for botrytis control, because the fungicides we have are so good against botrytis, although we're losing some of them. And never ever do it if you know you have anthracnose. And usually by this time of year we would know what plant supplies have anthracnose problems, and we would put that out. This year I've seen none, which is really good. But what happens with anthracnose, every time you touch a plant and then you touch the next one, you're moving that anthracnose pathogen. And so we get the worst disease where we do sanitation practices if there's anthracnose present. So, but again, we know what problems are with certain sources, and we try to get that information out. And this year I didn't have to put much out. It was a very quiet year. And then again, we want to initiate fungicide at first bloom, and we have specific programs that we recommend, and I'll come to that. The next disease is anthracnose, and there's it's can be really sad. You look at a field like that and you think, that's a great looking crop, and everything looks really clean. But then if you go into the canopy, all of a sudden with the right weather, you can have severe disease. And there's 3 pathogens, although we don't see this one much anymore. So mostly we talk about this Colototrum acutatum, the fungus, and it causes anthracnose fruit rot. It's very high in the southeast in terms of importance. The other one causes more of a crown rot, and it's low to moderate in terms of concern. Both of these typically come on the transplants, and the Gliosporoides, the crown rot, can come from native trees in our area. But what happens is they come on the transplants, the pathogen builds up with wind-driven rains, and it splashes from one plant to the other, and it also then can get on the berries and cause severe problems. And of course, I'll show some of the symptoms. So here is the anthracnose ripe fruit rot. You can imagine one spore landing on that berry, and then you get this round lesion, sunken lesion. And if it's wet, see that there's orange there? There's probably like a million or several million spores in there. So you can imagine one raindrop coming, bang, hitting that, and all those spores splashing to the neighboring berries. The crown rot, the pathogen gets in the crown, and you see this wilt. Often it's a sudden wilt. And if you cut open the crown, you have this marbling in the crown, this red and white pattern. And it's usually pretty easy to diagnose. Sometimes it's hard to tell it from Phytophthora, so usually I like to take it to the clinic, or if I have a microscope, I can scrape these roots here, and I often can tell if it's Phytophthora or not. But anyhow, you get a whole plant collapse. In the plug production facility, the acutatum strain, it can affect the young growth. And you'll see here, see that petiole? It's just rotting there. And here the young tissue is being affected. And again, I can usually take a microscope and scrape that tissue and get a rapid diagnosis. Sometimes, but rarely, if we have really wet weather during bloom and early fruit development, the strain can cause like this kind of flower or early fruit blight, or even on green fruit it can cause a dry lesion. The crown rot pathogen, it usually doesn't show too many symptoms on the leaves, but now and then you can have these black spots. The fruit rot pathogen doesn't affect the leaves usually, but it can affect the runners. You can see a lesion here on the runner. And so that's what we would be looking for if we're going into a field or people send us samples. This was a really bad problem several years ago. This is one grower, had 60,000 tips, right? And you can see that some of the tips were not too badly affected, others were severely affected. And if there's a lot of spores that wash down to the roots, and you get this root problem. And there were about, I think there were, well, there were quite a few million plants that went into Florida last fall that had this problem. And those plants were being stunted and or didn't perform well. So that was a serious problem. I don't often see that. What we do, again, it's the nature of this fungus. It lands on the green leaf and then just sits there. So you'll never see the first sign of it in a nursery, in many cases. Then you bring it to the southeast where we have lots of rain, lots of hot temperatures, and the pathogen becomes active. And then this one, it can be on a green leaf and produce spores and not show any symptoms. So it's spreading and building up and you don't know it. Or when the tissue dies, then it grows out. So what we do is we collect leaves and then we dip them in gramoxone, right, which is paraquat. Right, so that instantly kills the tissue. We surface sterilize it, dip them in paraquat, and then we rinse them off, and then we incubate them for about 5 to 7 days, and then the fungus grows out. Look at that. Those are called acervuli, but that's a Colletotricum acutatum. And look at it, this leaf is just full of spores. Okay, so that's one way we can detect the pathogen before it shows up in the fruit. So we're trying to, of course, develop technologies. You know, how quickly can we detect it? But you know, I've walked a lot of nursery fields. When you walk in a nursery field 10 acres, and there's probably maybe 250, 300,000 plants per acre they're going to harvest, what's the odds of me missing it, right? So that's a challenge. That's the challenge we face. It's not that the nursery growers are at fault. It's just it's not seen. We don't have the technology to detect it. And then if we detect it, most of my growers will say, well, you know what, since we've got anthracnose in the nursery plants, I'm not going to grow strawberries this year. Right? That's not going to happen. You've got the land ready, you've got it fumigated, and so you got to figure out how to minimize your risk. The best thing is to have clean plants. But you know, Travis asked an excellent question earlier, how do you know they're clean? Right? So that's a challenge. Okay. And that's a matter of practice and science. The other thing in North Carolina, and just like here, you know, this is a pine tree, 50 feet tall, Virginia creepers climbing up it. You know, and I had a grad student postdoc, they'd go up, you know, 40 feet, collect Virginia creeper leaves, paraquat them, and find Colletotricum gliosporoides. So it's in wild grape, it's in Virginia creeper, it's in oak. It has quite a broad host range, but the challenge is, and so this is actually, you know, so that pathogen comes from those trees, lands on the strawberries, we get complete plant wilt. And we can see, so we do inoculation studies, we can see some black spots, or we get this true wilt. But the problem is, we pick 10 wild strains of this fungus, and only 3 of them hit strawberry. So now we got a problem. We can find the fungus maybe, but it may or may not be the pathogen that affects strawberry. So again, that's a challenge. So that's, in terms of biology of anthracnose, we have the anthracnose fruit rot, anthracnose crown rot. You know, infested plants are the main source. Would be true here also. If you had anthracnose fruit rot last year, you will not get it again this year, unless you carry over some plants, because it has to live on green plants. But I've never seen it on a farm because they had it last year. I've seen it on a farm the second year in a row because they got dirty plants again. Okay, so in terms of rotation stuff, that's not important for this one. You know, sometimes we get infested tips, and of course then we're misting for what, 7-10 days, and then we slowly slow down the mist. After 4 weeks, we got 100% Calotropicum spread through those plug plants. And it comes in, you know, without showing symptoms. So those are the key parts about the biology of the organism. And then, you know, just the management techniques. I talked about them. Now, sometimes it works that if you find a little hot spot in your field, we've had success of just quarantining that portion of the field, pulling all the plants, and then bury or burn them. You can't just leave them in the field because it'll produce, the more they die, the more spores they produce, right? So you got to get them out. And if you're organic and you got anthracnose fruit rot, you have to do two things. One, you got to really pray for dry weather, and you have to take all the fruit out of the field. With Botrytis, you can leave it down. It'll pretty rapidly get colonized by other bugs, and Botrytis is so prevalent, you won't win that battle anyhow. So then again, we have specific programs, and I'll come back to that. So what we do is you know, we screen products, both biological products as well as fungicides, and then we ask the question, okay, this one controls anthracnose well, this one controls Botrytis, how are we going to rotate these into a program? And we have the other goal, is we don't want to use a fungicide too often, because then the fungus becomes resistant to it, right? So we want to use it wisely, steward it well. And so we put together IPM-based recommendations. There's quite a few different compounds that are out there and currently labeled, and this is just a brief list of them. And then, so the way we do our trials is, this is some Castlehane, and this would be a Botrytis trial. You can see what we do is we would spray this plot, and then we'd have an area in between. And then actually tomorrow, every Tuesday now, we harvest those plots. And now with anthracnose, you know, the splash dispersal, if this is a control, that's going to produce a lot of spores, and it's going to splash into our other treatments. So you'll see on some of my slides, we might have 25% fruit rot on the best treatments. And you say, Frank, I can't stand 25% fruit rot in my fields. But if you spray all your acres that way, you get like 2% or less. That's our experience. So if they perform well in our trials, then they can perform excellently in your fields, okay? This would be like Botrytis. This is the total number of fruit, and you can see how we get high pressure early in the season. Lots of fruit affected early when it's cool and wet. As it warms up into May, then Botrytis really tends to tone off. But just to give an example, this is something you should never do, but this is something we did. So this would have been early work, late 90s, early 2000s. We just spray elevate, elevate, elevate. Just switch, 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 right? You never do that, but you see how good control those products get. They're great Botryticides. The strobilurins, Pristine, Quadris, etc., are not. Well, the strobilurins themselves, Quadris and the strobilurin component of Pristine are not good fungicides. Pristine has another compound that is a good Botryticide. Then the other key part is you know, we could spray every week for 9 weeks and get pretty good control of Botrytis. So if we don't spray over the season, we get 6% on average. So now that's over the whole season, right? Now early in the season, the first week or two, we could have up to 20 to 30% fruit rot because of Botrytis. Like last week, we harvested for the first time. We had a lot of Botrytis where we didn't spray because it was such favorable weather. But you know, the whole month of May, we usually don't see a lot of Botrytis. So over the whole season, if we got 6% fruit rot, if we spray 9 times with a rotation program, we really reduce the amount of fruit rot. But see here, we put 4 sprays on during bloom. And that's as good as 9 sprays. So you don't, for Botrytis, you don't need to be spraying when your customers want to be in the field. You just need those 4 bloom sprays, bang, 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 and then you'll get good control. And then what we do is we take about 10 or 20 of our best berries we surface sterilize them in bleach, and then we rinse them off, and then we put them in these nice little plug trays. You see, if we don't spray, almost 100% of the fruit will show botrytis. So we pick perfectly healthy fruit, cut the calyx off, do those treatments, and then incubate them for 5 to 7 days. And this is George when we get wine in the lab. You know, it starts to smell like wine. You know, but look at this treatment here, 100% control of botrytis, because it stopped it in the flower stage. Okay. And we can put that to numbers, you know, about 85% fruit rot, right down to about 10% or less with certain of our rotation programs. So that's what we do, is we look in the field, we look in the lab, post-harvest, and then try to develop recommendations. Likewise for anthracnose, we can have heavy anthracnose pressure. This is number of fruit, and we see products here giving us very good control, especially those strobulurins. One thing that I need to say, if you do use fungicides, this is early work, 2002, and I just wanted to highlight 2 things here. One, in our early work, Quadris gave us excellent control. So if we didn't spray, we had 17.5% of the fruit rotted, and Quadris gave us really good control, as well as Pristine here. This is when Pristine was a numbered compound. So I just keep that thought for a moment. The other thing is, here you can see the sanitation. You know, we tended to aggravate our problems with anthracnose when we do sanitation work. The problem is, in the last few years, where we've used Quadris or Abound, here we had 67% fruit rot. So it was a heavy disease year. And Quadris is not working, nor Abound. Okay, so I don't know why. We've done a lot of work to try and figure out if the fungus is resistant or not, and it's not. Here, Pristine, you see, is working good. But see, that's a lot of disease. But if you did this program in your field, you'd get good control. And we just did this for scientific purposes to figure out what's going on, and we haven't got it figured out. We just know it fails. So that's why in our recommendations we don't put Abound or Quadris in our recommendations as a primary product. And this is what it looked like. Pristine gave great control. Abound and Quadris compromised control in the control. The other thing we do is, we've done a lot of work with this with Florida, South Carolina, Ohio, and Iowa, is, you know, to what extent can we predict when we need to spray? And so we have a field sensor that measures leaf wetness, or we can predict leaf wetness using mathematical algorithms through a company called Skybit. And what that is trying to predict, like anthracnose and botrytis are driven by temperature and moisture. And so we would only want to spray when there's a risk of a problem. So that's what we've done. We've got this forecasting model. And here, this is anthracnose fruit rot. In this year, this is 2011, had 8.5% fruit rot. If we spray every week, we got 3.2%. If we sprayed 3 times using the forecasting model, we got the same level of control. So, and you know, with the technology now, especially all this climate change data and stuff like that, you know, the technology to monitor weather patterns, predict weather patterns, and to interpolate between my farm and your farm, unless you live in the mountains, you know, we got hollers and hills, topography is tough. But I think we're going to come to a point where you can have a phone, a smartphone, and you'll be pushed data, say it's an anthracnose, you're in a high risk anthracnose time right now. You need to apply Cap10 or Pristine, depending if you're moderate risk or high risk. And so if you're flexible, then you can spray according to these forecasting recommendations. Some growers are not flexible. They say, well, you know what? Joe just sprays every Wednesday. That's his job, right? That's when he's got to get on the tractor, he's got to spray, because on Thursday he's doing this, and you know, so they do spray every week. So it depends on your farming operation. But that's where we like to head, right? We want to spray according to the biology. We want to spray according to the weather patterns. And most growers know this. They say, well, you know, botrytis, it's been quite dry. I don't have to worry about it. And so they'll stretch out their sprays. And so this is an example of that trial. This was last year or 2011, I forget. Here's the Anthracnose fruit rot, 80% fruit rot. Now see, you go from 0 to 80% fruit rot in less than 20 days. All right, so if it's there and if the weather favors it, it shuts down your shop. But here is the weekly spray program and the forecasting spray program. And again, you got to remember, we have a lot of pressure in that field. So normally our regular program would almost zero out that problem on a commercial farm. And we monitor the leaves of the pathogen leaves. We can have high levels in the leaves where we don't spray, and where we spray or use a forecasting model, we can reduce the amount of the pathogen in the whole field. So that's great stuff. All right, I'm going to skip through that. That bears out in fruit quality here versus this is cumulative marketable yield. If we don't spray, we lose a lot in marketable yield. This is harvest per plot. All right. The other thing, and I won't talk about this too much, we've been working a lot with our breeder who just left, so we're hoping to get another one. But you know, we want to always move ahead the sustainability of strawberry production. And one of the best ways to control diseases is by host genetics. So if you get the plants that don't get the disease, you're a step ahead, right? So that's going to be a challenge. And we've done a lot of work. You know, like Chandler's highly susceptible, but Bish is a variety that's been released, and here's a couple of North Carolina releases. Very low, keeps it, even without any sprays, less than 5% fruit rot. So it's very promising. All right, you're going to see more and more products coming on the market that are pre-mixes of fungicides. So like Pristine, it has a fungicide called Boscolid, and has a fungicide called pyraclostrobin. And this is the energy system of the plant. So these are like different complexes here in the energy system of the plant. So if you turn off one switch, let's say with pyraclostrobin, the fungus only needs one mutation to turn that switch back on. But if you turn off 2 switches, the fungus has got to do a lot more genetics to get a double mutation to overcome that resistance. So that's where these mixes are important, and or that's why we rotate chemistry to prevent the organisms from getting resistance. So if you go to smallfruits.org, we have our recommendations all spelled out for the Southeast. Every year we update them. We meet every year, and if you're an agent or whatever, you're welcome to come. We meet every year in Asheville, early part of March, and we hash out our recommendations for disease, insect, weed control recommendations. And then we put it in this publication, and then we post it on the web at smallfruits.org, which is located, it's hosted in Georgia, but it doesn't matter, it's for the whole Southeast. And we put efficacy tables in there, you know, what product works really well or not so well for what type of organisms. Okay, so just to help you make decisions. And then have very specific recommendations. With your first spray, do this. With your second spray, do this. Third spray, do this, etc. And again, you see what we're trying to do is we're trying to take, if you know you only have gray mold and you don't have anthracnose, you can save a lot of money. If you've got both, then we have a separate recommendation for that. You know, so again, it's all on that website, and it just gives you a nice basis to figure out how to implement a fungicide spray program. The organic market is not nearly as mature in terms of available products that give good disease control, where we can put these products together in, you know, in the recommendations. So, you know, we'd like to see it move that way, and I think it will, with biocontrols, etc. All right, so then finally I just want to talk about, you know, there's a bunch of kids, and they climbed up this mountain, and there's a wise man that's always up there in the mountain, and they said, "Oh, wise man," and he took this dove, and they said, "Oh, wise man, tell us, is this dove dead, or is this dove alive?" And of course, if he said, well, it's dead, they would have let it go, mocked him. And if he would have said, it's alive, they would have crushed it, and again mocked him. And he said, young men, the answer lies in your hands. Right? So, this is especially true with strawberries. If you overuse a fungicide, or if you overuse one type of method to control a pathogen, the answer is in your hands that that pathogen is going to shift. You're going to get genetic changes or a changed pathogen is going to come in that can overcome the resistance or the fungicide. And we're seeing a huge problem in the southeast. So normally, resistance, we would say, okay, what that is, that's normally a fungicide would work, and now it doesn't. That's what we would call resistance. You know, if you sprayed a tree with a fungicide, nothing would happen to the tree. It's naturally resistant to that fungicide, right? Because it's not a herbicide. So we're not talking about natural resistance. So that's what we're dealing with. And there's different levels of risk. So an organism that produces a lot of spores, and if you're trying to control it with a fungicide that only controls one switch, there's a high level of risk, like what we had with benlate on Botrytis, or Quadris on Anthracnose, or Elevate on Botrytis. High level of risk here. And I think it's kind of like a shotgun versus a sniper rifle, right? If you shoot a shotgun, that's like Captan. Lots of pellets, broad spectrum of activity, and it's hard to miss your target. But if you are a sniper, you get very precise shot. But if your target moves a little bit, you miss. So that's what these fungicides are like. Captan is a broad spectrum active ingredient where we've had no resistance reported on any pathogen, and it's used a lot. Whereas something like benlate, as soon as it came out, it didn't take long for a single mutation in one base pair to get resistance to that fungicide in a lot of different pathogens. So that's really what we're talking about. And we have two types of resistance. One is where we have a mutation and there's a sudden shift, or over time, the pathogen population becomes resistant. This is like benlate. This is work we did in our lab. Here's benlate. Here's the Botrytis growing on no benlate. Both of the strains are happy. Here's one growing at 1 milligram, which is about what we would apply in the field. Look at this one's completely controlled, 100% control. This one looks like it's eating benlate for lunch, right? It's growing really well. Here's at 100 times the concentration of the fungicide. This one is still growing. All right, so when we get resistance to some of these, it's a strong resistance. So this is work from Guido Schnabel. So what Guido does is any one of you can take Q-tips, collect 10 Q-tips of Botrytis spores off your berries or off your tissue, send it to Clemson, and he'll run a report about what your Botrytis profile looks like. And I'm not going to show all the slides, I'm just going to show a couple here to bring home the point. What he does is he puts them in these wells. And these wells have no fungicide or different concentrations of a fungicide. And then he takes your Q-tips, so you would collect them from the berries, takes your Q-tips, puts them in the well, and in a short time Botrytis grows or it doesn't. And then he'll know what fungicides work on your farm. And what's really interesting is every farm, most farms seem to have a different profile. Okay, so it's very helpful. So you can see here, pristine, this one's growing on pristine, that strain is not. You see, so we got resistance to the boscoli component of pristine. Here's Elevate, all right, sensitive strain, resistant strain, growing well on Elevate. So if you have a lot of Elevate resistance on your farm, you don't want to be using that product. Elevate's a great botryticide. That's the only thing it controls. It has no other value in your strawberry production system. So, and then he'll give you a report like this. You know, what kind of resistance profile you have and whether or not you should be using that product. Okay? And he's done a lot of surveying, including a bunch of samples from here. Okay? And here's just, I'll just give a couple examples. This is Iprodion or Rovrol. Rovrol is the product we would recommend for crown rot control. You can only use it once per season, and you have to use it before bloom. So when we have a winter with high crown rot, that's when we recommend that product. Let me go to another slide. Here's Boscovib, which is the active component of pristine. If it's red, that means more than 20% of the strains from that area are resistant to that product. Okay, you see a lot of red in these across different states. So that means that product is failing on a lot of farms in these different states. And here's the number of farms. Okay, so he sampled 10 farms from Maryland in 2012 and 2013. This is the switch. Switch is two products like Protonil and Fludiopsinil. Look at Fludiopsinil, no red. So switch works really well so far in most states. But don't use switch every week, right? That's the key. And I'm just going to dance through this. This is Elevate. Look at all the red. Arkansas, lots of red. North Carolina, where is it? Oh, not too bad. South Carolina, lots of red. Florida, 100% Elevate resistance in Florida. Okay. Thompson M, which used to be Benlate, all red. The other key, and you know what, I don't remember if we had this on our website, but Guido put this together. And every chemical has what's called a FRAC code, fungicide resistance code. And what we want to do is make sure we don't use fungicides in the same family back to back. We want to rotate them. And so this table is quite helpful. And if we don't have it on the website, I got to remind Guido that we should put it on. Let me stop there. That's Botrytis and Anthracnose. Those are the two main ones. I have some stuff on Phytophthora. I have some stuff on angular leaf spot. We don't have to cover that, but I just want to make sure we're all on the same page for Botrytis and Anthracnose, or if there's any questions about that. So if you're an organic grower, what can you do? One, buy disease-free plants, but I can't exactly tell you which plants are exactly completely free or not, you see? And do sanitation before bloom. Those are two critical things. Yeah.
In high tunnels, where we have prolonged, protracted bloom on those plants.
Mm-hmm.
In Botrytis is our main problem.
Yeah.
So, do you have any suggestions on how to go about and avoid or reduce the chance, the probability of resistance?
You know, I haven't worked as much in high tunnel strawberry resistance, but with tomatoes, two main things are sanitation. So remove all dead and dying tissue as soon as you can, and remove, you know, spent blossoms that didn't fruit and stuff like that. That's one thing. And then environment, which is hard in high tunnels because you've got no environmental control. You know, and at least in greenhouse tomatoes, we can vent, we can heat, and we can cool down, because the goal is to prevent dew formation. Just like this, you know, this bottle I'm holding. Look at all that dew formation. You know, there's high humidity air. The plant's cooler. You're going to get dew formation right on the leaf, which is perfect for that spore. In a high tunnel, you can't control that. Not too much, anyhow. So I think sanitation would be the main thing. And if in organics, you know, I don't have good recommendations for what bio products work well. You know, there's some trichodermas that are on the market that work well, and I don't know if people have experience here. You know, if you've used products or you've done things, but when it's cool and cloudy, it's a tough battle. You know,
Which was, I mean, this winter was cool winter.
Yeah, it was a tough winter for Botrytis. It really was. That's true. Yeah, Travis.
So when you get a resistant variety or whatever of Botrytis, so you're saying it constantly stays in your soil?
Yeah, it could stay in the soil because it does produce a black structure that stays in the soil. Although we've had a hard time finding those. We've done a fair amount of work looking in soils for it. But it probably can affect 200 different types of plants. So it's probably on the farm. So what this data almost says, it looks like it's a farm by farm problem. Yeah. Yeah, so that's a good question. That's what it looks like. Yeah.
What's your recommendation? Okay, so you went through, I'm using my farm for example, went through the series of rotating 4 sprays here in bloom. Okay, now I'm picking. I still got lots of blooms out there. What's kind of your thoughts on that for preventing botrytis?
Usually by that time, you're getting into warmer weather. So botrytis likes it. I'm thinking like a scientist here, 20 degrees Celsius, and so that's 70 or lower, cool and wet. That's kind of cool. Then we don't see a lot of botrytis later in the season. So that would be one immediate thought. The second thing is, if you got a lot of bloom, what I kind of think about is you got to think when your end harvest date is. You're about 26 days away from your last harvest. And so you might spray up to that last, where you think if you're usually going to hit June 5th or something like that. I wouldn't spray too much more than 26 days before that. So that's kind of another rule of thumb. Yeah. All right. So I stop there. We're pressed for time a little bit here.
The next thing was about the greenhouse.
Okay.
You guys, Phytophthora can be a problem in strawberry, especially in flooded. We had one year where we had flooded, a pipe busted. So
yeah.
We had a very susceptible cultivar too.
And angular leaf spot.
Yeah. Okay.
A problem too.
Okay.
Actually, that's sometimes coming from the nursery.
Yeah, it does.
Yeah.
Almost 100%.
Yeah.
Yeah. So this kind of looks like the Anthracnose crown rot, but it's all red. And a lot of times I can't tell, that's my job, right? I see these a lot. There's a few clues. When you pull the plant, just try to pull it. If it snaps, it's probably Phytophthora. If it doesn't snap, it's probably Colatochicum. But like it'll snap off right here where the rot is. So those are a few things, but you really should send it to a clinic to get diagnosed because the management practices are completely different. And what I do is I will take the roots and I'll scrape them, and then I'll see these round oospores in the roots. That's very diagnostic for that pathogen. So that's, if you bring me a sample, I can often tell within 10-15 minutes if it's one or the other. And they're water molds, so they like water. So I have this in the handout, and it's pretty clear. It's a lot of it is about water management. It's about land preparation. Although some of the worst problems we see with Phytophthora is on soil that was fumigated and then gets wet, because you kill the whole biological vacuum. You make a biological vacuum in Phytophthora. You have one organism, one food source. It can cause a lot of damage. All right, we talked about fumigation, so let me not hit that. Really, there are 2 fungicides that are available. One is Riddim Gold. You put 1 pint per treated acre. So if you have 2 acres of strawberries, you only have 1 acre under plastic. So you apply 1 pint per 2 physical acres, because you only got 1 acre on plants. So it's a very, very efficacious product for Phytophthora. And there's only been resistance reported in 1 farm in South Carolina. I haven't seen resistance anywhere else.
Does that just control it, or does it actually kill it?
You know what? It's fungistatic, so it controls it. And it only controls it in the plant. So that this chemical gets up in the plant, it actually gets into the plumbing system, and then it stops the Phytophthora. And it's good for 30 days or more. So that's way, if you know you have a problem, you apply it, but you got to think about another application within 30 days if it's the fall. Or if you know you got a problem, you can do it in the spring. And then what I like to think, I think I have a slide. Yeah. You know, so when do roots grow? The roots grow in the early fall. And then, you know, the plant tells you when it's starting to kick off in the spring. Well, when it kicks off in the spring, that's when you get this other rapid root growth. So we try to time the Ridamil applications according to the plant growth. So we want to get it, this is usually, here it would be probably mid-February. Or
So
late
it's going to be
February.
a continuous thing then, to try to control it.
Yeah, you know, that's a strange thing. Normally, Phytophthora, once it's in your soil, it can be a serious problem. But this one, only some farms do I see them have a recurrent problem. So I cannot say it's a continuous thing for most farms. Yeah, unless you grow Sweet Charlie. Sweet Charlie's really susceptible. Or what susceptible line did you have?
Radiance.
Radiance, it was really susceptible? Okay. So, yeah. The other thing is, with these phosphites, there's a bunch on the market. If you have poor root growth, or if you have a resistant population, these phosphites do pretty good. But they're not as good as Ridamil. So, I think that's enough to say. I have some other slides of data on that. And then diagnosis, you know, how do we tell them apart? I walk through that. And then angular leaf spot. So angular leaf spot can occur on a sporadic basis. It's always plant associated. It's a bacteria called Xanthomonas. It only can live in association with the host. Once the host is gone, the pathogen is gone. So you won't have it on your farm unless you carry over plants. And this is a severe case where the plants are really showing like they're stunted and they're pretty ragged. And the very diagnostic thing is you take a leaf and you pick it, and then you hold it to the sun and look at the back side. And these lesions here, they'll be translucent, so the light comes through. It's very diagnostic for this disease. Other leaf spots don't do that. What the problem is it usually doesn't affect yield, but look, if all your calyxes look like this, with the bacteria, it looks ugly. And pick your own is not as bad, but if you're trying to market them and all your calyxes are black, it really affects the look of your fruit. Again, we have monitor and manage, but the most important thing is, you know, there's a guy in Florida doing a lot of work with nurseries, trying to help nurseries make sure they don't have it. And what happens is in California, in their nurseries, they don't have the weather patterns that allow for it to be expressed, but the pathogen is building up. And then you put it in a wetter climate or a cooler climate, especially when we have to do frost protection. For some reason, that seems to really aggravate that disease. You can use copper, and I do recommend copper to our growers, maybe 3 sprays around bloom if it's cool and wet, to prevent that calyx phase. But, you know, we put on 11 copper sprays and didn't see control because the pathogen is in the tissue. So you're just trying to protect new tissue. And if you use too much copper, your plants can get damaged. But that's under hot conditions, and when we use 3, we don't see damage. But keep an eye on it if you're using copper. So that's about it, you know, for those 2 diseases. Those are the 2 other most common ones we see. Yeah.
Using Ridamil right after planting, is there a protocol for how many days or weeks you want to wait until you apply Ridamil, or can you do it right after planting?
Most of the time, it depends on how you plant the plants. So we did a lot of work with peppers. If the Ridamil's there when the plant goes in, that's the best. But you got to think about worker exposure issues. Okay. Plus, a lot of growers don't want to have the drip lines set up before they start planting because everything gets in the way. So the sooner you can apply it. So what, you know, you got to water in your plants, and then as soon as you feel like your plants are established and you're getting new root growth, that's when you want to put Ridamil in. Yeah. So you have to have active roots, and you're past all the establishment procedures. And then if there's a known problem, you probably would do it 30 days again, depending on what the weather's like. And then in the spring, you probably do 1 application, and if you've had heavy pressure, you could do it again. But usually that 1 application after transplanting, that one application during early plant growth does a really nice job. And Ridamil, even though it's systemic, you see strawberry fruit don't have stomates. So they only take in the water they need to size. So the Ridamil doesn't bioaccumulate in the fruit. It accumulates in the leaves. So I forget the days to harvest, but it's surprisingly short for a systemic fungicide. Yeah. Very good.
Are there any other questions?
I had one on the crown rot, because I had a strawberry just last night. I was out my high tunnel,
Yeah.
and one of my crowns, not the whole plant,
Okay.
just one crown, shoo,
Yeah.
was just laying there, just wilted right over. And I reached over and I grabbed it, and it just
Okay.
snapped right off.
Okay, because there's two things that would come to mind. One is sometimes, just the way the crown grows, sometimes they just have a poor connection with the mother crown. And they just kind of snap off when the fruit load comes on.
Okay, because it was loaded with berries.
Yeah, so it could be just a growth thing,
Okay.
or what you need to do is you'd have to cut that crown open, because that's how the Colletotricum crown rot shows up. It often shows up on one or two crowns first, and then you see just part of the plant wilting, crown by crown.
Okay.
So that's the other possibility. But the way you describe it sounds like to me, especially would you say it wilted pretty fast?
Mm-hmm.
Yeah. So because the Anthracnose one, you might notice it a couple days. The first day there's a leaf or two flagging, and at night they might pick up because then they're not transpiring as much. The next day the whole thing starts to flag, and the next day it's gone, kind of thing. But the way you describe it sounds it snapped.
Okay.
Sounds like it snapped off.
Okay.
It just had a poor physical connection.
Okay.
Yeah.
Is there any application for lime sulfur to control anthracnose in strawberry? Because I know we use it in grapes, blackberry, when it's dormant or when there's 1/16 inch bud break, and plants are already green when they come in. So
Yeah. I have no data on that. Offhand, I don't think so, because lime sulfur works really good for the mildews, which kind of grow on the surface of the tissue and all that. So just based on the chemistry and the biology, I doubt it, but I don't know for sure. Yeah. All right. Thank you.
Thank you. Well, first of all, I'd like to see just a show of hands. How many of you plan on being here tomorrow?